Yeah. Hi, everybody. It's a pleasure for me to present you today how pseudogenes and plasmid genomes reveal past transitions in plasmid mobility. So we know from the three domains of life that there are varying frequencies of pseudogenes among prokaryotes and eukaryotes. For example, eukaryotes may comprise a lot of pseudogenes, while many pseudogenes are rapidly depleted from prokaryotic genomes. And the general consensus is that prokaryotic genomes are usually devoid of pseudogenes. But there are also plasmids, and many prokaryotes carry plasmids, and they are genetic extrachromosomal elements. And they are known for being agents of horizontal gene transfer, as they are often self-transmissible via conjugation. Such plasmid traits like the plasmid mobility may explain why genetic alterations are different between plasmids and chromosomes. And therefore, we wanted to elucidate whether pseudogene evolution on plasmids follows different rules compared to chromosomes. And we have been subdividing this objective roughly into three parts. First, we wanted to quantify the pseudogene frequencies on plasmids and chromosomes. Secondly, we wanted to determine the gene functions that are commonly non-functionalized on plasmid genomes. And third, we have been following back the pseudogene evolution on specific plasmid lineages. For this study, we have been downloading plasmid-containing prokaryotic genomes from NCBI. And briefly, we have been using a taxonomically diverse dataset for describing general trends, and a dataset that was restricted to enterobacterial isolates for conducting robust and detailed analysis. The pseudogenes in the genomes have been annotated using NCBI's prokaryotic genome annotation pipeline and pseudofind, and afterwards we have been using comparative genomics and phylogenetics approaches to tackle those objectives. First, we have been comparing the pseudogene density of plasmids, here shown in orange, to the pseudogene density of chromosomes in purple. And indeed, we found a significantly higher pseudogene density in plasmids compared to chromosomes. To further pinpoint which plasmid traits might be associated with a pseudogene density, we have been classifying the plasmids into their mobility types, here shown as conjugative mobilizable and non-mobilizable plasmids. And indeed, we found an association of the plasmid mobility with a pseudogene density where non-conjugating plasmid showed the highest pseudogene density. Additionally, we also, on the genomic basis, we found also that there are a lot of transfer-related genes that are frequently non-functionalized in this plasmid genomes, and this overall indicated that the evolution of plasmid mobility might be linked to this frequent gene non-functionalization in plasmids. And to determine specific plasmid lineages, we have been calculating the pairwise shared gene content among plasmids, and darkly colored rectangles in this matrix here represent related plasmids with a high shared gene and pseudogene content. We have been analyzing adjacently located plasmid clusters in this matrix that have diverged, but likely share an evolutionary trajectory based on their shared gene content. One plasmid cluster comparison that I would like to explain here today very briefly includes the plasmid cluster E5, where the plasmids in this cluster are mainly non-mobilizable, and they are homologous to the virulent plasmid P0157 in enterohemorrhagic E. coli. And these plasmids seem to be related to the plasmids of the cluster Fe, and these plasmids correspond to conjugative plasmids, and they are diverged, and they are residing in Klebsiella, Salmonella, and Escherichia. This overall means that these two plasmid clusters represent related plasmid clusters with a shared genome content, albeit with alterations in their mobility class. And the varying percentages of the pseudogene content among those two plasmid clusters also indicated that the frequent gene non-functionalization here might be linked to the transition in this plasmid mobility that this plasmid cluster comparison showed. So we have been reconstructing the phylogeny of 316 plasmids included in those plasmid clusters Fe and plasmid cluster E5 based on 10 single copy genes. And you can see here that the phylogenetic branching pattern of the plasmids included in the plasmid cluster Fe is much stronger compared to the almost polytomic leaf of the non-mobile plasmids in cluster E5. And furthermore, the two plasmid clusters are separated on the phylogeny while the root split was distinctly determined in the conjugative plasmids. So this means that we afterwards concluded that the non-mobile plasmids included in the plasmid cluster E5 must be derived plasmids from the conjugative plasmid cluster Fe. And on the genomic basis, we were also able to show that the mechanism that was causing this is a large segmental deletion of transfer-related genes, followed by frequent gene non-functionalization in those plasmids. We have been also reconstructing the host phylogeny of Escherichia, Salmonella, and Klebsiella based on 133 chromosomal genes. And here you can also see that the three genera are separated on the phylogeny, which is visualized by this inner coloring. And afterwards, we have been mapping the plasmids from the plasmid phylogeny to the host phylogeny by using the color scheme of the outer coloring. And we have realized that indeed the conjugative plasmids are residing in diverged isolates of Escherichia, Klebsiella, and Salmonella, while in contrast, the non-mobile plasmids are residing in closely related E. coli strains, mainly corresponding to EHEC. And taken together, we have here the evolution from conjugative plasmids, so self-transmissible plasmids, to non-mobile plasmids residing in this distinct spot on this host phylogeny of those closely related strains, which is a signature of plasmid domestication. And plasmid domestication here means that these plasmids have become an integral genetic complement of this bacterial lineage. To briefly summarize, I showed you here that plasmids usually tend to have a higher pseudogene density compared to chromosomes. Additionally, I also showed you that there's a frequent gene non-functionalization of transfer-related genes and that this may lead to transitions in plasmid mobility. And in the end, I also emphasized that the loss of self-transmissibility is a possible route to domesticated plasmids. And with that, I appreciate that I've been accepted here for giving a talk about this recently published paper. I would like to thank my supervisor, Professor Tal Dagan, for the great supervision and for publishing this work with me, and my co-author, Dr. Yiging Wang, for contributing to this work. Thank you.
Yeah, we have a time for a couple of questions.
Not so much a question, but kind of a warning that NCBI recently changed their annotation pipeline. So anything you do with this, you have to make sure that you are using the same version throughout the whole analysis, like say same genomes and same pipeline, because they now they change the annotation for many pseudogenes have changed.
Yes, we are aware of that.
Okay,
that it's kind of inconsistent if you use genomes that have been annotated in different years. But that's also the reason why we've been using this second pseudogene annotation pipeline, which is called Pseudofinder.
Thank you.
Yep. Thank you.
I was curious about the sequence similarity, if you've looked at that for the domesticated plasmids or the ones that get these pseudogene changes as well. Do you think some of these pseudogenes might be acting as sponge RNAs, impacting the influence of other plasmids in the same bacterial cell?
The sequence similarity of what? Of
the pseudogenes
of the
themselves
pseudogenes itself?
between plasmids. So I'm curious if, like, in cases where a gene transitions into a pseudogene instead, if it's acting as a sponge RNA for other genes that might actually still be protein coding in other plasmids in the same cell?
Yeah, so we haven't checked anything that's related to RNA or the expression level itself, but what I can say is that after this shift from self-transmissible to non-mobile plasmids happened, that astonishingly these plasmids got very stable, and that the divergence of these plasmids was kind of reduced. So the pseudogenes did not diverge so much anymore.
Okay,
Compared to the conjugated plasmids, at least.
interesting. Thanks.